Good morning, students. This is myself, Dr. K. Shivaya, Department of Pharmacology, TVM College of Pharmacy. Our next topic is electron transport chain. So, in this electron transport chain, it is mainly oxidative phosphorylation takes place in the mitochondria. So, what is electron transport chain? The transfer of electrons from NADH plus H plus and FADH to oxygen via multiple carriers. That electrons combine with the oxygen and the energy released. So energy released from this oxidation reduction reaction used to derive the synthesis of ATP from ADP with combined with the phosphate. So that's why it is called as oxidative phosphorylation. The NADH and FADH2 undergoes oxidation. When it is undergoes oxidation, the movement of electrons happen. That electrons combined with the oxygen to produce the ATP synthesis. This transfer of electrons is done by multiple carrier which constitute the electron transport chain. So that this is ETC, the chemical reaction between electron donor and electron acceptor to the transfer of H plus ions across the membrane through a set of mediating biochemical reaction. These H plus ions are used to produce ATP, adenosine triphosphate. This is overall image how the ATP is synthesized in substrate level and oxidative phosphorylation. The electrons carried via NADH or electrons carried by NADH FADH. In the glycolysis, it is takes place in the cytosol. You know, the substrate level phosphorylation. That is example is glyceraldehyde 1,3 bis phosphate is converted into glyceraldehyde 3 phosphate. There one ATP is synthesized. That is the substrate level. There is no oxidation there. Substrate only to synthesize the one ATP. That's why it is called as substrate level phosphorylation. At the same time, in the mitochondria, in Krebs cycle also, one GTP is synthesized. Succinyl coenzyme A2 succinate when it is formed, one GTP that one also one ATP is synthesized. But in the glycolysis, we can produce this some NADH, and in Krebs cycle we can also produce the NADH and FADH. These NADH and FADH2s enter into the mitochondria. There it is involved in the oxidative phosphorylation. They transport the electrons through the electron transport. That electron transport oxidative phosphorylation happen in the takes place in the inner membrane of the mitochondria. That's why it undergoes oxidative. That's what it is called as oxidative phosphorylation. This is overall mechanism of the substrate phosphorylation and oxidative phosphorylation. I said already the multiple carrier are involved in the electron transport chain that are NAD and flavoprotein, nicotinamide adenine dinucleotide and flavoprotein, and non-heme metalloproteins are iron sulfur proteins. So these iron cycles between 3 plus and 2 plus both are states as there when compared with the hemoglobin and myoglobin. And one more carrier protein is coenzyme Q. Coenzyme Q. The Q it is denotes ubiquinone. So serves as anchor or to inner mitochondrial membrane, and cytochromes, electron transport proteins that contain heme. These cytochromes have the heme prosthetic group. But non-heme, this is iron. It is in both. It will come Fe3 plus and Fe2. It is interlinked changes happen. These components divided into 5 complex enzymes: complex 1 enzyme, complex 2 enzyme, complex 3, and complex 4, and complex 5. The first 4 complex enzymes are the transfer of electrons happen. In fifth, the complex 5, the ATP is synthesized. So the complex 1, simply we can note it as NADH dehydrogenase. So these NADH dehydrogenase have the 46, it is contains 46 proteins and prosthetics FMN, and it help to the that with the help of FMN and iron sulfur proteins. And complex 2, succinate to coenzyme Q reductase. Succinate reductase simply is NAD dehydrogenase. This is succinate. Here it is the number of, it contains number of these 5 proteins. It is mainly involved FAD and iron sulfate. And complex 3, transfer of electrons from coenzyme Q to cytochrome C. It have the number of these coenzyme Q. It consists of 11 proteins. So that transfer it involved the prosthetic. It has the prosthetic groups cytochrome C and iron sulfur proteins. Cytochrome 4, complex 4 enzyme have the cytochrome oxidase enzyme. It have the 13 proteins. It have the mainly cytochrome A and cytochrome A3. This is the how these complex enzymes and coenzymes transfer the electrons from NADH plus H plus to complex 5 and from FADH to complex 5. So there it is in complex 4 to synthesize the oxygen molecules, and what finally you can get the oxygen molecule and carbon dioxide and releases the ATP. See, if you observe here in complex 1, see in complex 1, transfer of electrons from NADH to coenzyme Q. Just use NADH2 coenzyme reductase. Otherwise, you have to write the NADH dehydrogenase enzyme. Just you write, better you write NADH2 coenzyme Q reductase, because of these NADH after releasing transfer of electrons from substrate to FMN, then after FMNH2 transfer the iron sulfate, then iron sulfate to transfer to the coenzyme. That's why NADH2 coenzyme Q. Okay, here the FMN involved between the transfer of electrons from NADH to coenzyme, FMN and iron sulfur. This is important. We have to write, explain how the electron transport from the NADH to coenzyme Q, that is NADH to FMNH2, FMNH to iron sulfur proteins coenzyme Q. That's all. Next, complex 2, succinate dehydrogenase. So this is otherwise you can write succinate to coenzyme Q. Why it is called reductase? Because of these are all these coenzyme Q reducted. This coenzyme Q reducted because of it taken the electrons. When the substrate loss of electrons, we can call as oxidation. When the substrate taken up the or gain the electrons, we can call as reduction. So these coenzyme Q transfer the electrons to the cytochrome B. So here the complex 3 next started. This complex 3 transfer the electrons from coenzyme Q to cytochrome C. That's why coenzymes to cytochrome C reductase. So in between we can observe cytochrome B, iron sulfur and cytochrome C1 and cytochrome C. And complex 4, cytochrome oxidase, cytochrome oxidase. Because of here the otherwise you can write the cytochrome A3 reductase, cytochrome A3 reductase. See here the complex 4, these cytochrome oxidase, cytochrome C, it is undergoes oxidation and A2 transfer of electrons from A to cytochrome A3. Then these electrons are transferred to the oxygen, these transferred oxygen molecules and the loss of hydrogen molecules. Okay. So loss of hydrogen molecules, loss of electrons, we can call as oxidation. So these hydrogen molecules move, and electron molecules also, hydrogen molecules and electrons move from NADH plus H plus to FADH2. From these hydrogen molecules and the electrons both are transferred to these oxygen molecules and to form the water, and these electrons, here the hydrogen is transferred, the electrons utilized the H plus ions, that here it is the electrons utilized by the complex 5 and synthesize the ATP from the ADP plus phosphate in the F1 and F0 particles. You know, already well studied about the mitochondria structure. It have the outer membrane and inner membrane. The inner membrane have the folded, that folded we can call cristae. That cristae, on the cristae we can observe the F0 and F1 particles. So what you studied and what I discussed in complex 1, complex 2 and complex 3 and complex 4. So complex 1, NADH2 coenzyme Q, complex 2, succinate coenzyme Q reductase, complex 3, cytochrome reductase, complex 4, cytochrome. Why it is called cytochrome C oxidase? Because I here completely lost the, here the final cytochrome C reducted, but here the cytochrome C lost the electrons and added to the oxygen molecules, and many of the components are proteins with prosthetic groups to move electrons. So what have we discussed? Same, the complex one, NADH dehydrogenase. So for again it has a 46. So the movement of electrons NADH2 coenzyme Q. So here it is involved in FM and flavin mononucleotide. Okay, NADH plus H plus. Here NADH plus H plus. It lost the electrons to FMN. Here then FMN form the FMNH2. Then FMNH2 loss. Then again to form this. Excuse me. So these iron sulfur molecules transfer the electrons to ubiquinone or coenzyme Q. So finally the coenzyme Q to form the QH2 along with the electrons. So what is the main about ubiquinone? It is a benzoquinol linked to a number of isoprene units. It is a 3 redox states. Fully oxidized means ubiquinone Q, partially oxidized semiquinone, fully reduced ubiquinone. See here, this is ubiquinone because of fully reduced, because of the transfer of hydrogen, these NADH plus H plus, these H plus ions transferred to these coenzyme to form the ubiquinone. Okay, so one electron along with have the electrons, H plus ions and electrons. And complex 2, succinate dehydrogenase, entry point is FADH2. Okay, so already see here FADH2 is not here, it is not FMN. This is FAD. FAD. See here the FAD is enough. Okay, so here the transfer of electrons from FADH to coenzyme Q. Here the enzyme, why it is called succinate dehydrogenase enzyme? Why it is called succinate? Because of the succinate to converted it to fumarate. Succinate to fumarate, when it is converted during the time only, the FAD is converted into FADH2. Already we studied in citric acid cycle. So these FADH2 transfer the electrons and the hydrogen molecules, iron sulfur cluster and ubiquinone. Now it is why it is called ubiquinone? Because of QH2. See, succinate of the Krebs cycle is also called complex 2 succinate. So that's why succinate to fumarate to form the FAD to FADH2. So this is the how the FADH2 formed. So now yielding to produce the QH2 because of fully saturated. See, fully reduced. So when it is fully reduced, we can call as ubiquinone. Fully reduced because of the coenzyme Q, it is converted into QH2. So it does not pump any proton during the transport of electrons across the inner material. This is not important. You have to understand the transfer of electrons and hydrogen molecules are very important. Next, complex 3, cytochrome B, C1 and C. These three are involved in the cytochrome C. So in complex 3 accepts electron from coenzyme QH2. Already the first, we discussed. So once again, I'll show you. C is the cytochrome B to cytochrome C. Now this is cytochrome coenzyme Q. From the coenzyme Q to receive the cytochrome B and cytochrome C1 and cytochrome C. So in complex 3, which components are the cytochrome B, cytochrome C1 and cytochrome C? These components are involved. Okay. So the QH2, so the combined it releases the two protons into the transmembrane space. Within complex 3, the released electrons are transferred to a iron sulfur center and then to B type cytochrome or cytochrome C1. See, that image is very important. You have to depend on that image, you have to write the matter. Okay. So here we have to talk about cytochromes. Cytochromes, only cytochromes. Cytochromes are proteins with heme prosthetic group. Heme prosthetic group. It carries electrons one at a time to complex 4. So they are mainly transfer the electrons from first and second to 4. From the first complex to 4 and the second complex to 4. So the major respiratory cytochromes B, C, A. So to in that have the cytochrome, they have the, again it is subclassified the A and A3 and B, B1, B2 and C, C1, C. So cytochrome C is a key regulator of programmed cell death in a mammalian cells. And then complex 4. See, it is have the only A and A3. It is having only A and A3. So 10 protein subunits, 2 types of prosthetic groups and 2 heme and 2 copper molecules. Always you see, just remember this chart is very, very important. If you remember the C, complex 3 have the cytochrome B, cytochrome C1. Now complex 4 have the A and A3. That's all. Then transfer of electrons from cytochrome A to A3, and transfer of hydrogen molecule from cytochrome A to A3, and attached to the oxygen molecule to produce the water. On the basis we have to write the matter, and the basis only I prepared these all things. Okay, got it? So electrons delivered from cytochromes A and E3 to oxygen. Several chemicals can inhibit the pathway at the different locations. Cyanide and carbon monoxide can block the transport between the A and E3. This is for the inhibitors for the oxidative phosphorylation. So cyanide and carbon monoxide to block the transport between the A and A3 and oxygen. And complex, same. So it catalyzes the transfer electrons from C to oxygen mass. It contains the okay already state. Then 3 copper ions arranged as 2 copper centers. So designed as copper A and copper B. So copper A contain 2 copper ions linked to by 2 bridging disulfide residue, and copper B is coordinated by 3 histidine residues. So this is a structure of that complex 4 enzyme. Okay, cytochrome oxidase structure. It and also contains the copper A and copper B. So the 2 protons per pair of electron are pumped across the membrane, and another 2 protons are transferred to molecular oxygen to form water. Okay. So here the metal centers of cytochrome oxidase in complex for heme A and heme A3. So 2 heme, 2 adjacent QB. So this is the how this is the structure of complex 4 enzyme. Electrons enters complex 4 one at a time from cytochrome C to copper A. Then they pass via cytochrome A to the binuclear center where the chemical reaction takes place. This is the how the electrons transfer. If you are confused the entire matter, don't just simply write the electron transfer from cytochrome C to copper A. Then after that copper A to last electrons and transfer to the cytochrome A. Then after that cytochrome A to transfer electrons cytochrome A3 are adjacent copper B. So then after these electrons are transferred to the oxygen molecules and also it releases the hydrogen molecules to produce the water. Like this way we can write. If you confuse here, this is I am discussing about only structure, but the major important point here how the electron transfer. This is the way of a transfer of electrons. Complex 5. This is the last step for the synthesis of ATP. The mitochondrial ATP synthase here consists of 2 multiple subunits. Here the enzyme is ATP synthase. That ATP synthase enzyme have the 2 components, 2 complex F0 and F1. See, this is F0 and F1. So F0 is an electrically driven motor that spans the lipid bilayer, forming a channel through which protons can cross the membrane. F0 provides a channel for proton. See here it provides the transfer of these protons, into the here. It provides. So F1 harvests the free energy derived from proton movement down the electrochemical gradient by catalyzing the synthesis of ATP. F1 phosphorylate the ATP to ATP. So this is the after gradient this proton gradient came, these H plus and this H plus when it came, then the ADP bind with the phosphate to form the ATP. So this is the ATP. So this is the complex of ATP synthase structure. So some 3 to 4 hypothesis is there for the how the ATP is synthesized. That a well known and exhibited, that is chemiosmotic hypothesis. So proposed by Peter Mitchell in 1961. So this hypothesis coupled electron transport to ATP generation. Michael says ATP generator use the energy in the form of a proton gradient across the biological membrane by direct chemical transfer of high energy groups. What he said, the protons transferred to the mitochondrial membrane through the F0 and F1 particles and to synthesize the ATP. So how these that is transferred? Complex 1 and 4 appear as a proton pump. Complex 1 and 4 appears as proton pump which transport the protons across the membrane due to conformational changes induced by the electron transfer. In complex 3, protons are carried across the membrane by ubiquinone. Complex 1 and 3 pump 4 protons per pair of electrons. Complex 4 pumps 2 electrons per pair of electrons transported, and other 2 protons are combined with the oxygen to form water. See, just this is the how flow of electrons. This is a complex 1, this is complex 2. So these are from NADH, from NADH plus H plus is converted into that loss of protons and electrons and give to the these Q through the FMN and iron sulfur. So then it is enter into the complex 3 directly. It is enter into the complex 3. Then here again the complex 2 here also to transfer the electrons to the Q ubiquinone. It is transferred in cytochrome Q. So these coenzyme Q, though so not cytochromes, coenzyme Q, these coenzyme ubiquinone receive the electrons in the complex. These complex 3 and transfer the electrons to complex 4. In the complex 4, utilizing the electrons and H plus ions to produce the so off photon 2H plus ions to produce the water. See how these proton pumps in happen in the electrons. So that is the electron transport. This is a proton transport. See the complex 1 transport the 4H plus ions. So here NADH is converted into. So this is the transfer the 2 electrons and 4 H plus ions to here. Then here the electron transfer to here. Then after that here the transfer of hydrogen molecules to coenzyme Q. Then coenzyme Q it is in the form of I said coenzyme. This is coenzyme Q ubiquinone. When it is this is full reduced. This is the endoplasmic reticulum. This is cytosol. This is inner membrane. This is inner membrane. So when it is electron transfer, the 4 hydrogen molecules transferred here. Then so that but here we have to remember the main important point is the transfer of electrons from complex 1 to complex 3, transfer of electrons from complex 2 to complex 3, and movement of hydrogen ions. Just you write it, the movement of ions from NADH plus H plus 2 directly coenzyme Q and FADH2 to coenzyme Q, FADH2 to coenzyme Q. So here then after that the electrons transfer cytochrome C, then cytochrome C2, cytochrome A and cytochrome B. So already we discussed. Then finally the ATP is synthesized here. This is imaginated diagram how the protons are pumped here inner This is matrix. This is cytosol. This is endoplasmic radius. Okay. This is the complex 1. This is the complex 3. This is the complex 4. How they are involved structurally. Then finally the H plus ions, when it is pumped, then ADP is converted into ATP. This is the ATP synthase complex. The functions of electron transport chain. So increasing the redox potential because of oxidation and reduction, both are takes place. That's why it is increased reduction potential. Decreasing the electron pressure because of when the electron transport, automatically decreasing the pressure. Diminishing the free energy levels, it produces the free energy. So along the chain, there would be step by step flow of electrons from the most negative initial donor to most positive terminal acceptor site. See, the electron movement from NADH2 or FADH2 to transfer to finally oxygen only. Am I correct or not? So transfer the electrons from NADH plus H plus to FADH2 to oxygen. So here, then electrons entering to electron transport chain are energy rich because of to produce the energy. So much of this energy get conserved in ATP form. So in electron transport chain, enzyme bound H plus is used as the fuel for the energy generation. It's available from the dehydrogenation reaction of substrate oxidation. From H plus ions donating substrates, H atoms are collected by dehydrogenase and supplied to electron transport chain through the NAD and FAD. We studied in Krebs cycle and citric acid cycle, are nothing but citric acid cycle and glycolysis. So they are serves as acceptor and carrier and a donor of H plus ions. The H atoms soon donate their into electron transport chain. The H plus ions soon escape to the aqueous medium. So here the overall the discussion part, the redox substrate of NAD plus and oxidized to form the NADH plus. So these NADH plus are utilized in the electron transport chain. From the coenzyme charted electrons are funneled to molecular oxygen, though chain of electron carrier. They include flavoproteins, that includes flavoproteins, iron sulfur molecules, ubiquinone, coenzyme Q, and cytochromes. And respiratory electron does for 3 ways as electron sulfur, as hydrogen atoms, and hydride ions. So whatever it is, these are all components involved in the electron transport chain. These are all only discussion what already we discussed. So overall, oxidative phosphorylation, the electron transport chain moves electron from NADH and FADH to oxygen. In the meantime, ADP phosphorylated to ATP. This is we can call a simple definition for the oxidative phosphorylation. The transfer of electrons from NADH and FADH to oxygen. In the meantime, to produce the ATP. So 3 ATPs are generated. 3 ATPs are generated when 2 electrons are transported from NADH to oxygen. When 2 electrons are transferred from NADH to oxygen to produce the 3 ATPs. The oxidation of FADH2 only produces the 2 ATP. If you take an FADH2, transfer the 2, we can synthesize the 2 ATP. This is regarding the electron transport chain. Thank you.